Theory of evolution, modern synthesis. When Darwin published his theory of evolution and his findings, at the time, findings by Gregor Mendel describing the rules of inheritance hadn't really gotten that much attention. But many years later, when his discoveries came to light, scientists realize that they can really express in evolution in a whole new way in terms of through the lens of genetics, and that gave them a really powerful tool to actually quantitate evolution. So this video describes what that means. Darwin discovered and described the concept of descent with modification, which basically said that we inherit characteristics from our ancestors, and those characteristics that we inherit change over time because the environment changes. But one thing that Darwin didn't know was how these characteristics are inherited. Then Mendel discovered how these rules of inheritance work, and that allowed scientists to create the modern way to describe theory of evolution. So which we call modern synthesis, which is the connection between genetics and evolution. And the modern synthesis looks at evolution at two different levels. At a micro level, it talks about evolution within a population. So how evolutionary pressure can gradually change the genetic makeup of a population. We call this microevolution. So microevolution looks at one population at a time. However, at a macro level, answering the question of how new species form, we called it macroevolution. So the idea is that when we talk about successive microevolution, events happening in a population that eventually leads to formation of new species, which is called macroevolution. So if we describe it in a visual form with respect to microevolution, for example, we look at the evolution of this population of these beetles, and only these beetles. And then once this beetle evolves and once this population experience number of different microevolution happens, then new species arrive. So when we talk about macroevolution, we talk about not only changes within, let's say, if we talk about dinosaurs, this population of dinosaurs, but as you accumulate enough changes, eventually you get different species. So how is the modern way of expressing evolution? Let's look at microevolution first. It is expressed as allele frequency. So if you remember, the allele is different versions of a particular gene. So in population genetic terms, evolution is defined as a change in the frequency of an allele in a population over time. So, and as an example, let's say if we think about the human population, if we look at the different alleles in the blood type system in human beings, there are three alleles: the A allele, the B allele, and the O allele. So for scientists to take a look and see, okay, is a particular human population evolving with respect to the blood type system, you look at the frequency of different alleles. And the way you define the frequency of allele in a population, scientists look at the total number of alleles for different alleles for each gene. You add them up together, that's total number of all the alleles in the population. And then you can look at individual alleles and calculate the frequency. So the frequency of the A allele would be the total number of alleles for A, total number of A alleles divided by the total number of alleles in the population multiplied by 100. So as a representative, let's say this is a human population, and for this particular population, the percentage of A alleles is 26.1, B alleles is 30.4, and O alleles is 60.5. So when evolutionary biologists ask, is the human population evolving with respect to the gene that defines blood type, then the question becomes, is the percentage of these alleles changing or not? If it is changing over time, then that population is evolving with respect to this gene. Now, the entire total composition of the alleles and relative percentages for a population is called the population gene pool. So what would it look like in terms of, what would evolution look like in terms of allele frequency? So let's say we have this initial population and you have these percentages for different alleles. You look at this population for generations over time and you see that the percentage of alleles hasn't changed. Another situation is that you start with the same population with these percentages, but if you look at generations over time, you see that the percentage of these alleles have changed. So the question is, which one of these populations have evolved with respect to this gene? Did the top population evolve with respect to this gene? The answer is no, because the allele frequencies have not changed. What about the population in the bottom? The answer is yes, it's because the percentages of these alleles have changed over time. So that's the modern way of looking at evolution in a population. So the next question is, which is a question pondered by two mathematicians independently, Hardy and Weinberg. They asked, are there any conditions under which the gene pool in a population does not change at all, where the population does not evolve? And there are various different factors can determine whether the allele frequency changes. Not the only, natural selection is only one way that allele frequency can change. There are multiple different ways that this change can happen. And these are the different conditions. If there is no natural selection, if there is no mutation, mutation gives rise to the variation that is present in a population. If there is no migration, meaning that the gene pool on this side of the river, for example, remains separate from this side of the river, so the allele frequency doesn't really change. When you have a really large population, because when you have a really large population, even if there are small changes in allele frequency happens, in the grand scheme of things, it really doesn't make much of a difference. And mating has to be random. That means there cannot be any way that organisms, especially in sexual reproduction, organisms that choose their mate, there would not use any criteria to select one particular type of mate versus the other. So it's totally random. So therefore, we know natural selection causes evolution because it selects for certain characteristics. Mutations are the basic building block requirement for that leads to new variations, new characteristics. Migrations causes what's called gene flow, which means alleles from one population can travel to another population and disrupt that population gene pool. A small population is more vulnerable to chance events. And then non-random mating changes the genotype frequency, because if it's non-random, then only certain organisms, only certain members of a population with certain characteristics would have a better chance of reproducing and passing their genes to the next generation. So let's take a look at each and of these conditions. With natural selection, we already talked about the conditions that Darwin described with these Galapagos Island finches on Galapagos Islands, and the fact that differences in the conditions on the island gives rise to different percentage of birds having a certain size of beak. Now, natural selection can cause different end results, which we call it different types of selection: stabilizing selection, directional selection, and diversifying selection. So this all assumes that we're starting with a population that has similar representatives or a certain normal distribution of characteristics in the population. But then due to natural selection, the middle characteristic, it becomes reinforced and it becomes more common. And the example that we're seeing here is the number of eggs that is laid by a bird. And natural selection selects for a certain ideal number of eggs. Why is that? Because if the bird produces not enough eggs, because eggs, some of them don't develop, or maybe a snake comes and eats the eggs, if the bird doesn't lay enough eggs, then the gene for that bird doesn't go to the next generation of birds. Now, if you have too many eggs, then that bird is going to have a hard time going out and finding food for the new babies that are born. So if you have too many eggs, it will be hard to feed them. So those babies don't survive. So nature selects for this ideal number of eggs, which leaves a cushion for some of the eggs not developing or being devoured by a snake, and being conservative enough to numbers such that the bird can go out and bring food and help the birds to survive. The next situation is when the population diversifies and then only one extreme end of a phenotype is encouraged. So this is something that was observed actually during the Industrial Revolution. When Industrial Revolution happened, before it happened, there were these population of moths which had white areas and black areas. You had some moths that had totally black covering. You had some minority of moths that had totally no spotting, and most of their coloring was white. And then you have in the middle here, most of the members of the population of moths would have spots on them. But then Industrial Revolution happened, and then the area in which these moths lived all collected soot, which resulted into moths that had black covering to kind of hide from predators and camouflage. And the moths that did not have this all dark covering were easier detectable by predators, and they were eaten. So therefore, the environment that was created because of Industrial Revolution shifted the population phenotype towards one extreme end. And that's why we call it directional selection. It shifted the selection towards one end. Finally, diversifying selection gives rise to encouragement of the extreme ends of a phenotype. And this is a hypothetical population where you have a population of rabbits. Kind of most of them have a total white covering. Some of them had dark, and some of them had kind of a gray and some dark areas. And for some reason, some environment changes, and the most common type and the intermediate phenotype, which is the white covering for rabbits, make them vulnerable to being hunted. Then the only ones that could hide from predators would be the ones that are either dark or white with dark spots, and therefore you reinforce the two extreme ends of the phenotype. So these are three different types of natural selection. You should know what they are and need to be able to recognize examples of them. Another event that changes the allele frequency and leads to evolution is called genetic drift. Genetic drift is basically due to chance events. which is represented here. So we have a population of rabbits. The frequency of the A allele, let's say there are two different alleles for coat color, A, which is brown, which is the dominant, and little a is recessive. The population starts with equal frequencies for both of these alleles. But for some chance event, because of some chance event, only the rabbits that have at least one big A allele are able to reproduce. And then that changes the allele frequency for the big allele to go up and for the little allele to go down. And for the next generation, the rabbits with just homozygous A would be able to reproduce, and then basically the allele with the little a symbol basically disappears from the population. So this entirely due to chance. There is no adaptation or any selective pressure involved, but just some event happens and leads to the change in the frequency of alleles over time. Now, genetic drift, which are chance events, come in two flavors as seen in nature. One is the bottleneck effect, which means that something happens, an event happens that creates a bottleneck randomly for certain members of a population. So if you think about it, you have a bottle of these beads, and when you try to get some of these beads out of this bottle, only a few of them have the chance to get out and survive. Now, examples of a bottleneck are plenty, especially in the human populations, because human race is the worst kind of bottleneck that has been in nature. And one of the examples is the way we've been hunting the population of seals. So if this red line shows human beings to start slaughtering and hunting these seals, you can see before this slaughtering events, the population size of seals were pretty stable. But because of this bottleneck of human beings ravaging this population of seals, their population collapsed. And that's when we got our act together and made the hunting of them illegal, so therefore the population could recover. But if we hadn't, they would have gone extinct. Another example is this chicken prairie. When colonists came into certain areas of the United States and started setting up farms, that was the habitat for these birds. Their population really went down drastically, which the population of these birds almost disappeared. And this happens all the time in nature, and usually the culprit is the human race. Another type of genetic drift, which is a random event which leads to change in allele frequency in the population, is called founder effect. So on the left-hand side, you can think of a population of human beings, and let's say they have a variation, let's say in eye color. So we got green and blue, light blue and dark blue and brown. But for whatever reason, a small part of this population picks up and then goes to another place and becomes a founder of a new population. Now, since new population is taking a small sampling of a large population, it doesn't include all the different alleles that are present. So therefore, the new founder population doesn't have all the alleles in the original population. So therefore, the allele frequency changes, and this population has evolved. An example of a founder effect is the preponderance of this disease called Ellis Van Creveld syndrome, which is common among the Amish community, which traveled from Germany to the United States. So those Amish communities, they usually they interbreed within themselves. They don't mix with the rest of the community, but they did come from a larger German population in which the alleles for this particular, that causes this particular disease, wasn't really that common. But when this small population that had this allele common in between them came to the United States and stayed within themselves and formed a community for themselves, that led to a change in the allele frequency of this community, and basically it's a new population that has evolved. Notice that evolution doesn't always mean better. So there's a wrong impression that evolution always means we're getting better. But actually that doesn't mean. It just means that things are different. Gene flow is another way of changing the allele frequency in a population. Is just due to migration. So if you had two populations of these beetles living in different areas, and if some of these brown beetles come in contact with the green ones and they mate, and then that causes a change in the allele frequency of this population. Mutation is basically the building block of the variation in our population. If it wasn't for mutations, really evolution couldn't really happen, because there wouldn't be any changes among us. So an example of it is development of antibiotic resistance in bacteria. We have an original population of bacteria, some of which randomly developed a mutation. Now, if we put this population of bacteria in a new environment, which has antibiotics, over time, only the bacteria that have the mutation that make them resistant to the antibiotic are able to survive. So therefore, this population of bacteria evolves over time. So actually, in this case, you're looking at a combination of two things: mutations and natural selection. So there are a number of questions in here that are multiple choice that I will place at the end of this slide presentation. Make sure you attempt it